The goal of my research is to explore the role of a gene called ZNRF3 in adrenal cancer, looking specifically at how it may be involved in an intracellular pathway known as non-canonical Wnt signaling. The adrenal gland is a small, triangular-shaped endocrine gland situated above the kidney, and it is comprised of two distinct regions, the medulla and the cortex, which each produce important hormones for your body to function, including those important for your body's fight or flight response and stress response. Adrenocortical carcinoma, or ACC, is a rare malignant cancer of the adrenal gland that is aggressive, difficult to detect, and usually has poor prognosis, creating a great need to identify new potential therapeutic targets. My research is based upon findings that alterations in the gene ZNRF3 were present in cases of ACC, and patients with these alterations showed decreased overall survival. Therefore, one way to find potential new treatment targets is by understanding the way that the protein that this ZNRF3 gene encodes for interacts with other proteins to control cellular functions in adrenal cancer cells. These are known as cellular signaling pathways. Wnt signaling is one type of cellular signaling pathway, characterized in both development and cancer in animals. My gene of interest, ZNRF3, is known to play an important role in canonical, or the first characterized form of Wnt signaling in the adrenal gland. But we also hypothesize that ZNRF3 may influence non-canonical or lesser characterized forms of Wnt pathways. This hypothesis is based on several previous observations. First, while gain-of-function mutations that lead to activation of canonical Wnt signaling are found in both malignant and benign adrenal tumors, ZNRF3 alterations only occur in malignant tumors. Furthermore, ZNRF3 is expressed in both regions of the adrenal cortex where canonical signaling is present, as well as regions where canonical signaling is not. As a starting point for my research, I am using a gene editing technology called CRISPR-Cas9 in order to mutate the gene ZNRF3 and therefore knock out its function as a protein in an adrenal cancer cell line. I will then assess how this affects levels of other proteins in order to characterize its involvement in non-canonical Wnt signaling pathways.